Okay, here we go. I'm actually in class right now recording. This is so fun. Okay, I hope you've all had your coffee. We are starting Molecules of Life, Part 1. Oh, yeah. So just think of that name, Molecules of Life. These are molecules that all living organisms need. So let's start with the whole macromolecule concept. So macro means large. You could see that in your notes. Pull out your notes. Poly means many. So when we're talking about these macromolecules, we're talking about these huge molecules. And you're going to start with individual mono means one. So we're going to make these polymers, these really large molecules from individual monomers. Okay, so these are big words. So monomers are going to be used to make polymers. And they have to get linked together. They have to get stuck together. And so how they're stuck together is by these covalent bonds, which are strong bonds. So they're very strong bonds, the strongest bond. And so how they get bonded together is through this dehydration synthesis. So in the word synthesis means to make. Dehydrate, what would that be? What is hydrate? It's water. So dehydration synthesis, we're going to make something by removing a water. We're going to make the bond. So if we look at all the individual monomers, here's a monomer, and these are all bonded together. You can see those are bonded together. And then there's another monomer that wants to come in here and link up to this polymer. And you can see this HOH, which is water, H2O, 2 hydrogens, 1 oxygen. So we're going to remove that water, dehydrate, and we're going to form a bond. And so we're going to make this long chain, this polymer, by removing water. It's called dehydration synthesis. So think of the term synthesis means to make, dehydrate is water. So tear apart that word. That's how we're going to make these large polymers, these macromolecules. So, and then, let's go back here real quick. And then hydrolysis, to lyse, means to break apart or to split. So hydrolysis, lyse means to break apart, and then hydro is water. So with dehydration synthesis, we made a bond by dehydrating. And lysis, we're going to break a bond by adding water. So here's our dehydration synthesis, here's our hydrolysis. So now we have a bond. We want to break that bond. We're going to add water. We're going to split that bond, and then you can see that water now is attached. So now we have an individual monomer. If we wanted to keep splitting it, we'd add a water here and add a water here. So this is hydrolysis. Lysis means to break apart. Okay. Now, for these reactions, another thing that you need to know is that enzymes, their job is to speed up the rate of a reaction. We're going to go into a lot more details when we look at proteins on that, but just know that enzymes will speed up the rate of the reaction. Okay, so there's 4 main macromolecules that we'll talk about. The first one is a carbohydrate. This one, the prefix carbo is referring to carbon, and hydrate is referring to water. And so carbohydrate, basically the structure is CH2O. So carbon and water. And it's in that molecular formula. So you can see carbon, hydrogen, oxygen, carbon, hydrogen, oxygen. And you can see it's in a 1 to 2 to 1 ratio because you've got C, 1 carbon, 2 hydrogens, 1 oxygen. You just multiply it by the number of carbons, all of these. So it's in this structural format, a 1 to 2 to 1 ratio. So the carbohydrate, these are the individual monomers, and then we stick them together, and we're going to make these very large sugars. So they're mainly sugars. We have monosaccharides, mono meaning one. And I want you to circle or highlight that sac, S-A-C-C-H, that means sugar. So in your notes, circle that. So with a term like monosaccharide, you put those two prefixes together and we have one sugar. And so these are our individual sugars. You do not have to memorize what each one of these are, but just so you can see structurally the difference. So this one's 3 carbons. You can see there's 3 carbons, and then you're going to have 6 hydrogens, 3 oxygens. This one is a pentose sugar. It's got 5 carbons. This one has 6, and they're all in this type of format. A disaccharide, di means 2. That's going to be 2 monosaccharides that are bonded together. And what was that type of bond? What bond did we say what process would create a bond? That's right. Oh my gosh, I heard you answer it. Dehydration synthesis. I'm sure. And it's so funny because I'm recording this and I actually have the 7 Eclipse students in the class. So, and they're all watching another video. So, all right. So disaccharide, you just made a covalent bond by dehydration synthesis. Poly means many. So we're going to make these big, gigantic macromolecules, these big carbohydrates, by linking them together. And again, you have a 1 to 2 to 1 ratio. Another thing that you need to know, and I want you to circle this in your notes under 2C, it says the names usually end in "-ose". So when you see that, that's also a little nutrition tip when you look at nutrition labels and you're thinking you're not eating any sugar, but when you see galactose or high fructose corn syrup or ribose or any of the oses, that's telling you that that's a sugar. And it's these gigantic polysaccharides, these huge molecules, that are the primary energy source for cells. So carbohydrates can also be an energy source of stored energy too. So it's not just usable energy, it's stored. So for starches, that's the stored plant sugars. And then glycogen is the stored animal sugars. And so in animals, it's stored through glycogen. And then in plants, it is with starches. Now, cellulose is this gigantic structure. It's huge and it's really, really tough. And this is the component of plant cell walls. That's why plants are so tough and hardy. They have these cell walls. And it's this cellulose that is the structure of the cell wall. Very, very hard to digest. And then we have chitin, and this is the exoskeleton of some animals and also some fungi cell walls. And so we'll go into a little more detail with that coming up. All right, now let's move on to the second one. So our first macromolecule is a carbohydrate. The second one is a lipid. And the categories that are included in the lipids are fats, oils, waxes, and steroids. Most of them are hydrophobic, which means they fear water. Hydro is water, phobic means to fear, and they don't like water. They don't mix. You put them together and they're not going to mix. It's just like, going to a bad party or something. It's just not a good mix. Gosh, can you go to a bad party? I hope not. Don't ever go to a bad party. Leave if you go to a bad party. All right. So lipids are mainly composed of these hydrocarbons. And again, break apart the word hydrocarbon. It's got water and carbon. And all of the bonded hydrogens cause more energy to be released when they're broken off. So once these bonds are broken, which is how energy is released, period. And we have these, what we call triglycerides. So here's the structure of the fatty acid, is you have this glycerol molecule, and then you have these 3 fatty acid tails. And so triglyceride, 1, 2, 3, you have glycerol and Then the difference between saturated and unsaturated. Saturated fats, one long chain, Unsaturated has this double bond right here, so you have these bending that takes place in them. When we talk about the phospholipid bilayer, this phospholipid structure, it's got a phosphate group and it's got these fatty acid tails here as part of the phospholipid bilayer. And like I said, here's the bilayer, here's the phosphate group and the fatty acid tails. They're hydrophobic, so they fold inward because there's water here and water here, and they don't like the water. But the phosphate heads love the water, and they love partying with the water, so they are going to face the water. Waxes. These are, we combine alcohol and unsaturated oils. Lipstick, yay, is an example of this. Very important. Well, maybe not for all of us. And then the structure of steroids. So these are the components. These are steroids. Cholesterol is the type of steroid molecule. And the cholesterol is embedded in the phospholipid bilayer, and it provides for a fluid movement in that flexibility in the cell membrane. And then we also have hormones in this category, and those are used as communication molecules. Lipids are stored in adipose tissue, so fat tissue, and it can lead to hardening of the artery. So you can see here's what a normal one looks like, and then this one with all that fat built up. So don't eat those burgers anymore. Not good at all. Okay, that's all we have. Oh my goodness, we are done. I can't find where to stop it though. There it is.